Envelope, cell envelope. Okay. Cell envelope is a big topic. So what about cell? Let's break it down, make it more specific. Cell envelope. Yeah. Capsule. Okay. Glycocalyx. Okay. What about glycocalyx? Okay. No. You want me to just go over everything about glycocalyx?
Discuss it.
Sure. No problem. Okay, let's. Glycocalyx or capsule. Bacteria. Okay. They may have 3 layers. Okay. All bacteria, they have the innermost layer, this one, called cell, plasma, or cytoplasmic membrane. Membrane, right? Second layer, that many bacteria, or I'll say all bacteria, except all bacteria, except mycoplasma, is the cell wall. And the outermost that many bacteria have is capsule or glycocalyx. Capsule or glycocalyx. Okay. 3 types of glycocalyx. Slime layer, loosely attached to the bacteria, unorganized, loosely attached. 2, macro capsule, firmly attached, organized, firmly attached to the capsule. And then micro. These are the 3 types of capsules. Functions, what does it do? Okay. Give you 4 functions of this, right? The most significant function of this is prevents phagocytosis. Phagocytosis makes bacteria more virulent, pathogenic by doing that, right? Also, because it is mucousy gelatinous, attachment. Because it's extramucousy layer, also bacteria can survive longer. Okay, prevents or allows or saves bacteria from desiccation. Saves from desiccation. And finally, source of nutrition. Source of nutrition.
Capsules are made up of two walls, the cell wall and the cell membrane.
Nope. Each membrane is on its own entity. They're separate layers.
So that's what makes up a bacteria.
Yep. Okay.
Well, the cell wall and the cell plasma makes up the bacteria.
Okay. This is one bacteria. Okay. Let me write this down. Okay. This is Klebsiella pneumoniae. This bacteria looks like this. Okay. Or it could be Streptococcus mutans. No, Streptococcus mutans will be round because that's round shape. That's Klebsiella pneumoniae. Okay.
Looks like an egg.
Mm-hmm.
It's all in the center, and then you have the protein that feeds it and the protection on the outside.
Right. Exactly. This bacterium right here. No cell wall at all. No capsule at all. This bacterium is Mycoplasma. So it has only one layer. Which one? I should have used red color just to show you. I'm going to use red color, sorry. So you can see. Yeah, in plasma membrane only. So, okay. Yeah, just this layer. That's it. So this is mycoplasma. Mycoplasma. That's it. Now here's another bacteria that has this layer and this layer. Can you name this bacteria? E. coli, very good. E. coli. All right. Let's go. Capsule. Chemical composition of capsule. What is capsule made up of? Okay. That's it. 3 combinations. Sugar, which is polysaccharide. Sugar. Complex sugar. Okay, number 2. Protein. Protein. Or both. Glycoprotein. Glycoprotein. Okay.
Which is called
Sugar and protein. Sugar, glycoprotein combination. And important thing, names of bacteria that make capsule. I gave you, I believe, 3 examples or 4, I don't know. 1, second was Streptococcus mutans, and the third one was Streptococcus pyogenes. Streptococcus pyogenes. I think that's all we talked about the capsule.
When you say it saves from desiccation,
Dryness.
it's harmful.
Dryness. Dryness. Prevents from dryness. Yeah. Yeah. Drying. Yes.
I just wanted to go to osmotic lysis and osmotic collapse.
Oh, yeah. We are coming to second layer and then we'll talk about that. Sure, sure. Yeah. Coming to second layer. We'll do that now. Sure. Any question on capsule?
So that the slime layer was loose, so micro is tight. The macro is tight. Is the micro also tight?
Micro? Yes, it is. Yeah. Firmly attached. Very firmly attached. Yeah. So now let's go to the second layer of the envelope, cell wall. Cell wall. Cell wall all bacteria except mycoplasma, they have cell wall. Okay. First, let me make you a list. Okay, before we do anything else. Okay. Cell or cells, structures that do not have cell wall. No cell wall. no cell wall. Okay. Number 1, mycoplasma.
The one that causes pneumonia is Streptococcus pneumoniae or Flagella?
Both. I did not give you the example of Streptococcus pneumoniae, but you are right. It does cause pneumonia. There are other bacteria that also cause pneumonia. Okay, there are hundreds of causes of pneumonia, but I only told you one.
And then she has a she has a capsule, right?
Yes, it does.
And then Streptococcus also does, right?
Yes, yeah. Streptococcus pneumoniae, there are different strains of Streptococcus pneumoniae. Some have capsule, others don't. Not all Streptococcus pneumoniae, they have capsule.
Because the ones that have cannot cause the pneumonia.
Right. Streptococcus pneumoniae that have capsule, they are pathogenic. Streptococcus pneumoniae that do not have capsule, they are non-pathogenic. Okay? So mycoplasma, no cell wall. Number 2, L forms, mutants. Okay? Number 3, protoplasts. Remember, these are gram-positive bacteria that have lost the cell wall, right? Gram-positive bacteria that have lost their cell wall because of chemical treatment. And number 4, spheroplast. Partial. SP. Spheroplast are gram-negative bacteria that have partially lost cell wall. The outer membrane only. No, I'm sorry. The peptidoglycan only, but not the outer membrane. There? Peptidoglycan only, but not the outer membrane. If I draw it for you, okay? Here is the cell membrane. Both have the cell membrane, okay? This is the protoplast, gram-negative bacteria. They have what? But the outer membrane. They have the outer membrane, but they have lost their peptidoglycan. Yeah. They have outer membrane, but they have lost peptidoglycan. So both gram-positive and negative, they have plasma membrane. But spheroplasts, they also have outer membrane. Outer membrane. Outer membrane. Okay. Anything else that does not have cell wall? That's about it, right? Okay. Animal cells. Animal cells. Animal cells, yeah. Number 5, animal cells. Yeah. Animal cells. Animal cells. Protozoans, yeah. Protozoans have cell wall-like structures, but that is not considered true cell walls. Okay? Yeah. So, but we are going to stick with these 5. Okay? So remember these for the test. All of these, we are going to put it on one side, and everything else, they have cell walls. So what type of cells have cell wall? Have cell wall. Plants, fungi, okay. Green algae, all bacteria, except mycoplasma. Okay, that's it. Okay. Now let's talk about these bacteria. Okay. I don't know if the camera is covering. Who's good with camera? Someone needs to take a close shot of these so people who are not here can look. Anybody good with camera? Not want to take a chance? You want to take a, okay, go ahead. Go ahead, anybody. Oh, not with that camera. Sorry. Yeah. So, yeah, all right. So now we are going to talk about the gram-positive and negative. Maybe I should just do it right here. Okay. Gram-positive and gram-negative. Gram-positive cell wall, gram-negative cell wall. Okay. Pardon me? Yep. Mm-hmm. Peptidoglycan. 1, 2. Teichoic acid. Teichoic acid, okay. It's of course two types. Wall and lipo. 3, outer membrane. Outer membrane. I haven't told you yet who has what, so I'm just listing it. Okay? Then I'll put positive, negative. Who has what? Membrane, and outer membrane is made up of 4 things. 4, yeah. Phospholipids. Phospholipid. Porine proteins. Lipopolysaccharide. And lipoprotein. These are your cell wall. Who has what? Okay. Gram-positive, they have peptidoglycan? Yes. But they have more. Gram-negative, they have peptidoglycan? Yes. But less. Teichoic acid, gram-positive have? Yes. Gram-negative? No. Outer membrane, gram-positive? No. Gram-negative? Yes. The end. That's it. Yeah, really? Yeah. That's it. Simple. Yep. Yep. No need to make diagram. If you want diagram, you can make diagram if you want to. Yeah. E. coli is gram-negative. Yeah. Now we talk about hypotonic, isotonic. Okay. How do cells react in isotonic, hypotonic solution? How does the cell wall protect them or not protect them? This time I'm going to put in here a mycoplasma, each test tube, a mycoplasma in a plant cell. One mycoplasma plant cell. Okay. Mycoplasma and plant cell. Could be your bacterial cell too. Important point is that cell wall. This one has cell wall. No cell wall there, only what? Yeah, plasma membrane or cell membrane. Cell membrane. Okay. What's this in the middle?
Bacteria don't have nucleus. The DNA. Just DNA.
Very good. And what is that? Very good. So, yeah, see, tricking me. Yeah, that's a plant cell, that's a eukaryote, so that's a nucleus. Okay. All right. All right, good. Now, let's make the conditions. Isotonic in the middle one. Isotonic. Most isotonic environment for most living cells. How much salt should there be? 0.85 or 0.9%, 0.85, 0.85%. So sodium chloride. That is isotonic. Okay. Now, if I want to make it hypotonic, anything below this, anything below 0.65, that will be, yeah. Salt concentration, solute concentration should be lower than this. That will be hypotonic. If the solute concentration is 0.75, that will also be hypo. Okay, hypotonic. Hypotonic. Okay, so I'll just make it 0.5% sodium chloride, salt. Okay. Hypo. Hyper. Yeah. Pardon me? More what? More salt, exactly. Hypertonic. More solute. Less water, less solvent. Okay. First environment, more solvent, less solute. Okay. Now, osmosis. Osmosis, water moves from higher to lower, right? Yeah. So, who has more water, cell or the test tube? Test tube has more water. Has more. Exactly. So, water will rush in. Water is going to go in. Yeah. Water is going to go in. Okay. They both will gain water. Who has protection? Cell. The cell with the cell wall. So the cell will gain water. Okay. The membrane will be pushed right next to the cell wall, like this. Right. How about this one right here? It will gain water and eventually it ruptures. Right? That's why it's called ghost cell. Ghost cell, ruptured cell. And this is called plasmoptized. Tized, like a mop, soaks up the water but doesn't rupture. Plasmoptized. Yeah. Where should I write? Maybe here. Plasmoptized. Plasmoptized. Soak up water. This is a term for those cells that have cell wall. They gain water but don't rupture. Okay, cells with the cell wall gain water in hypotonic solution but do not rupture. Okay, cells with cell wall gain water but don't rupture. Isotonic solution, no change, no change, no change. Both cells remain or maintain their shape. No change. No change. Hyper tonic solution. Water will come inside where? In the tube, yes. So both cells will lose water because cell has more water. What will come out? Okay. Which one will lose shape? No cell wall. Mycoplasma. That is crenation. Okay, I'll just redraw the thing. That's how it's going to look like. Cell membrane shrinks it, but the cell wall maintains the shape. The whole plant cell will be empty. Yeah. That is called plasmolysis. Plasmolysis. Plasmo from cytoplasm. Plasmolysis. Cytoplasm breaks down. Why? It loses water. Remember, we just learned today 80% is water. When the water is gone, plasmolysis, cytoplasm breaks down. Cytoplasm, lysis, plasmolysis, not cell lysis. Cytoplasm breaks down. Okay?
Return back to its normal state.
Absolutely. Yeah. Remember, the cell is metabolically inactive. When you put it in isotonic solution, it will grow back. Okay? Yeah. Better? Very good. Sure. What's next?
Can you go into the, back to the capsule?
Oh, sure. Okay.
With the staining?
Staining. Okay. Yeah.
You know, and even like to go as further as to discuss negative stains?
Sure. Okay. All right. Okay. I just want to answer this correctly, so I want to make sure I understand. What is your actual question, though?
Like,
So,
over
the staining,
the staining of the capsule,
staining of the capsule.
reasons why
Oh, okay. All right. So, okay, let's take the.
we would not take a stain.
Oh, why would it not take the stain? All right, sure. All right. So here's the. Okay. Here's a slide. Okay. Okay. Capsule. Bacterium. Bacterium. Okay. So you're trying to stain it, right? Okay. You cannot stain the capsule. All right. First, what you need to do is stain the background. So you need to flood your slide with an anionic stain, such as nigrosin. All right. So you put nigrosin. What will nigrosin do?
It's going to stain.
Slide. So the whole background is going to be black. And the color of the bacteria will be what? Clear? Okay. Because bacteria is negatively charged? nigrosin is negatively charged. So both bacteria and capsule, because capsule is non-ionic. Non-ionic. Bacteria is negatively charged. And dye is negatively charged. So they will repel each other. So bacteria is clear, capsule is clear. There's no contrast, so we're going to have a problem. So now you can add any dye, any cationic dye. Once the nigrosin is dry, completely dry, then you can flood your slide with safranin, crystal violet, methylene blue. Now, let's say if I put safranin, okay, flood the slide with safranin. Bacteria will be what color? Pink or red. Here's your capsule stain. That's it.
Okay, there was a, I know you spoke about the differences because, say for example, in a negative stain,
Yep.
it can be, it's basically the same as a dark field.
Yes.
But there were some similarities, I think one similarity and a difference.
Yes.
Can you go over that?
Simple stain and negative stain. Yeah. Similarity between simple stain and negative stain is that they both use one stain. Both negative stain uses anionic stain, simple uses cationic stain. Yeah.
And then you say that under a dark field microscope? You see the negative stain under a dark field microscope?
No. Similarity. Similarity is they both use one stain. That's the similarity. They both use one stain. That's the similarity. Difference is different charge stain. One uses anionic, the other one uses cationic stain. Okay? Was that the question? All right. What next? Chapter 4, are we on chapter 4, 3, any chapter? Or questions from pre-test actually? Yep.
The question you had on Endospore, can you see Endospore under
Bright field microscope?
the microscope?
Under which microscope?
That's the question.
Oh,
Can you see living
yeah. Pardon me? Yeah, sure. Actually, I promise that I will give you a handout. I think Tamika asked me. I have a handout. I should have brought that handout. Okay. But, yeah, I can give you that handout. I probably should take a 5-minute break and should go get that handout. Yeah. I have a handout that one of my students made. I'll give you a photocopy. It tells you which microscope is for what purpose. Yeah. Not. Who said that? And then you woke up. No. Okay. All right. No. Okay. All right. All righty. Let me explain endospores. All right. Question is how to observe endospores, right? Okay. All right. You have a slide. You have endospores on it. You have vegetative cells on them. Okay. I think the question that I asked when we were doing bright field microscope, okay. Remember, endospores are inside the bacteria, okay, once they are made. I think the question that I asked when we were doing stains, I think, can you observe endospore under bright field microscope? That's the question I asked, I believe. Can you observe endospore under bright field microscope? That was my question. Yes. And yeah. And then I asked you the second question, can you observe endospores of living cells under bright field microscope? No. Phase contact. That was the difference.
What is the difference? The endospore, the living cell, and then the only were endospore.
Okay. Endospores of living cells, okay, you cannot go inside a living cell under bright field microscope. That's the difference. You cannot go inside a living cell, under bright field. Only phase contrast or confocal microscopes allow you to do that. That's the difference. But you can see endospore under bright field microscope. That's what you did this week. You stained the endospore, didn't you? Malachite green, and you saw them.
You just can see the inside.
Exactly. Yeah.
You can see
Yeah.
the outside on the bright field.
Of course, yeah. Yeah, sure. Yeah. So, okay. Not the intra. You can see intracellular structure.
It's just living that you cannot see.
Exactly. Can you see the nuclear material? Nuclear material is intracellular. Of course you can see, but it's not living. Not living, that's the difference.
And then so then you have to use phase contact.
Exactly. Exactly. Intracellular living and bright field. No, no, no. Intracellular living and bright field cannot go in hand. Okay? Yeah. Mm-hmm.
If I had say, okay, the capsule is growing in favorable conditions, the
Yes.
things is good.
Yes.
Okay, I have a vegetative cell. So, in good conditions, so it's going to grow a capsule.
Yes.
What?
No, wait.
You can't say, okay, you don't know the question I'm asking.
I'm not question. I meant if it's vegetative cells, it grows endospores?
I didn't say Endospore, I said a capsule.
It already has a capsule, why would it grow another capsule?
Go ahead. It's okay. Sorry.
You want to finish my question?
Go ahead.
I was asking a question.
She's just asking a question. Why do you have such a-
Let her ask question.
Okay, so it's favorable condition.
Yes.
And it has a capsule. Right?
Yes.
It's heat sensitive.
Yes.
If I want to see, I want to be able to see the inside of it.
Yes.
So I'm going to do a negative stain first to get the background, and then to stain it with a...
If you put a nigrosane on a slide, that slide is done. You cannot do anything else with that slide. If you have any leftover bacteria, you may go ahead and observe it under phase contrast microscope. Yeah, sure. Once you put a nigrosane dye on it, you can't observe that bacteria under dark field microscope. No. Okay. Okay. Yeah. You can observe it under phase contrast, okay, if you do not put any stain on it. Once you put the stain on it, it's done.
Right, because then it's black. Then it's black
Yeah.
on the background and it's
Right,
black.
right. No contrast. Correct. Yeah. Other topic. Yeah. Yeah. Yeah.
Can you explain just the part about acid fasting when if something is missing?
Sure. Acid fast missing. Okay. All right. Sure. No problem. Sure. Let's do that. All right. Okay. Acid fast. Acid fast. So acid fast, we are going to take acid-fast bacteria. We are going to take gram-positive. We're going to take gram-negative. Okay. First reagent is carbonyl fusion. Carbonyl fusion. About 5 minutes. Steam. Wash. Color of acid-fast bacteria. Magenta. Positive. Magenta. Negative. Magenta. Okay. Decolorize with acid alcohol. Acid fast. Magenta. Some of you over decolorized your slides today. Yeah, you did. And they were blue. Okay. So, yeah, they were not magenta. So, normally, if you do it properly, it's supposed to be magenta. Magenta. Positive. Negative. Methylene blue. Magenta, blue, blue. That's normal. Blue, blue. We are going to change something here. We are going to change acid alcohol with ethyl alcohol. Ethyl alcohol. Ethyl alcohol. Magenta. Any change here?
No.
Because this one is much stronger. Okay. So this one will remain magenta. Gram positive. This one will be magenta. Not strong enough. Magenta.
Clear.
Clear. Too much lipid. Clear. Clear. Gram negative. Remember gram stain? Gram stain. Ethyl alcohol removes the primary color. Okay. Clear. Yeah. Gram negative bacteria have too much lipid, and ethyl alcohol removes the primary color from gram negative. So that's why it's clear. So when you apply methylene blue, magenta remains magenta, magenta remains magenta, and now clear will become blue. That's why we use acid alcohol in acid-fast stain, so we will be able to differentiate between gram-positive and acid-fast. We do not use acid alcohol, we use acid alcohol. Okay?
So if we use the ethyl alcohol and then afterwards when we put the methylene blue,
Yep.
the gram-positive is,
Oh, this will be magenta.
Yeah.
Yeah, this will be magenta. Yeah, exactly. This will be magenta. Magenta. Magenta and blue. Blue. Those are. Yep. So it can remove the color from both gram positive and negative. Ethyl alcohol is not strong enough to remove the color from gram-positive. Okay?
You want to see the non-acid fast.
Right. Okay. Was this the question?
Yes.
Okay. All right. Okay.
So acid alcohol is strong.
Acid alcohol is stronger. Acid alcohol. Ethyl is not strong enough. Yeah. Okay. Now, gram stained. Everybody okay with the oxochrome missing and chromophore missing? Okay. One more time? All right. Sure. Yeah. Let's quickly one more time. Well, I had some papers in my hand when I came in. No. Did I put them in the back or I left them outside? No, 1 second. Oh. I have a sign-up sheet, if you please. Yeah. Okay. All right. Gram stain. Gram positive. My department chair asked me to keep a record, so, but I may at the end of the term. Yeah. Gram positive, gram negative. Okay, gram negative. A couple of students left, right? Okay, yeah, I remember their name. Okay. Okay. Old, gram, positive. Why am I drawing so big? I have no clue. Okay. Fine. All right. So your 4 reagents: crystal violet, primary stain, grams iodine, which is mordant, 95% ethyl alcohol, decolorizer, and counter stain, safranin. You add your crystal violet. The color of all 3 bacteria will be what? Purple, purple, purple. After grams iodine. Okay. Dark purple. Dark purple. Dark purple. Here comes the most crucial step. Gram negative. Clear. Clear. Positive. Purple. Old gram positive. Clear, we haven't added red or pink yet. Alrighty. Now we add saffron in. Gram negative will be red or pink. Red / pink. Purple. Gram positive remains purple. Old gram positive, red or pink. Red or pink. This is normal. These are your gram variable, also called gram variable. Okay. Now let's part these old gram positive. These. Yeah, these are also called gram variable. Okay. Yeah. Now let's play with it and make some changes. Okay. Crystal violet. Missing oxochrome. Okay. Okay. If it is missing, what is the color of this bacteria? Color of this bacteria. Okay. At the end. At the end. At the end. Yeah.
I'll be pink because no. Oh yeah, I'll be pink because that still has a pink. Yeah, it has oxygen.
At the end, what's the color of this bacteria? Very good. Pink or red. Very good. Because second is not missing anything. So it's pink or red. Very good. Very good. Everybody got it? Yeah. Primary is missing, so the stain is not there. So it's going to be clear. So let me write it down. It's going to be clear here, right? It's clear here. Clear here. And then it's going to pick up red or pink here. Got it? Okay, very good. All right. Now, if oxochrome is missing here, what's the color of this bacteria? At the end. Thank you. Everyone signed it? What's the color of this bacteria?
Clear.
At the end.
There's no pigment, so it's still clear. Oh, I'll be pink at the end.
At the end.
Pink or red.
Pink or red?
Saffron.
Yes, very good. Pink or red. Same thing, because gram negative, they always pick up secondary stain. So pink or red. Okay. So, this time, saffronin missing oxochrome. I didn't ask you. Oxochrome. What's the color of gram positive? Very good. Clear. Okay. Clear here. Clear. 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 clear? Clear? All right. Okay. Good. Good. So I think you guys got it now. Good. Good. Good.
So even, even in the all-gram positive, even if you add crystal violet, it will be clear.
Because all gram-positive, they do not hold on. Even nothing is missing from crystal violet, they don't hold on to the purple color.
Oh, no.
Because, yeah, because they lose their purple color here. You see it's clear?
Right.
They cannot hold on to the purple color. They become clear.
Is it because the
Okay,
glycan is
Yeah, it's not strong enough. Yeah.
but it started, like, when you put the crystal violet, the first step, then it's still clear, because you say that the all-gram positive won't hold there.
No, after, if I say, I think this was my first question on the quiz 1, pop quiz 1. What is the color of all gram-positive bacteria after the primary color? Purple. That was the question on the first. Purple. Exactly, purple after primary stain.
After primary, then after gram-positive, dark purple.
Purple.
And then after AD.
If I ask you what is the color of gram variable bacteria at the conclusion of gram stain? Red. At the conclusion of gram stain, it's red.
If nothing is missing.
Yes, yeah, red or pink. See, because they cannot hold on to the primary color. Okay. Yeah. What next? Fermentation? Sure. Fermentation is an aerobic process First of all, fermentation is a process in which cells make The purpose of fermentation is to change one organic compound into another organic compound, okay? And it doesn't need oxygen. It can take place in the presence of oxygen, but it doesn't require oxygen, okay? Starts with one organic compound such as sugar, and it is converted into another organic compound, okay? I gave you 3 end products, okay? Depending on the biocatalyst, okay, the organism, which organism is involved, it can be converted into different end products, okay? If the end product is yeast, if the catalyst is yeast, if the microorganism is yeast, you take sugar in a test tube, put yeast in it, it is going to turn it into alcohol plus CO2. Okay, that's one type of fermentation, one organic compound into another. If you take the bacteria Lactobacillus bulgaricus, this is going to make lactic acid, lactobacillus, lactic acid or acid. That's another type of fermentation. Okay. And E. coli, it can do both. Right? It can make acid, and it can also produce alcohol plus CO2, like that.
I thought you said it would be acid and CO2. And
Oh, that's right.
alcohol after.
Acid plus, absolutely. No, I remember. Not with, yeah, alcohol plus CO2. Yeah. Yes. Right. First, it will always do this and then this.
But not at the same time.
Not at the same time. One enzyme system at a time. Yeah, not both. Yeah. Yeah. So that's fermentation. Yeah. Pasteurization is you take these products right here, alcohol, acid, okay, and you heat them. Okay, could be juice, could be milk, could be alcohol, and you heat them at a certain temperature. Okay, the purpose of pasteurization is to kill certain types of bacteria. Okay, pasteurization does not accomplish sterilization. Bacteria, for example, bacteria that can cause TB, Mycobacterium tuberculosis, bacteria, Salmonella, Coxiella. So some types of bacteria, but not all. That is pasteurization. Heat treatment. Okay. The most common type of pasteurization is LTHT. Okay. HTST, not LT, HTST, LTHT, well, slipping. Okay. Yeah, high temperature, short time, 72 degrees for 15 seconds. 15 seconds, yeah.
Okay, there is one question on the pretest.
Pretest, sure.
That I want to know. Why?
Okay. All right.
This question, if you add 4 cups of distilled water to a 2-gallon fresh water fish
Sure.
tank,
Yep,
in which lived a group of happy bacteria metabolically active,
sure.
what would happen to the water concentration inside the bacterial
Yes.
cells?
Yep.
And,
And your answer is?
and the answer from the key, it would go up.
Okay.
Water inside.
Yep. Okay. Here is question number 4 on pretest. Here is a fish tank in which you have a group of happy bacteria. Okay. Now, what will happen if you add, these are happy bacteria, meaning what? They have, it is isotonic to start with. Now you add 4 cups of distilled water, meaning you are creating what type of environment? Hypotonic. Okay. Hypotonic solution, what happens to the cell? They gain water, lose water? Gain water. So they are going to swell. They are going to gain water. Gain water. Yep. They will be plasmoptized. The water constant inside the cell is going to go up. Yep. I don't know how to make the eyes go like that. I don't know. Yeah. Okay. Now what if I add 4 cups of salt? What will happen to them? They will?
No, it is hypotonic, now you'll make it isotonic.
Salt. So what type of environment I'm creating? Hyper. So the proper term will be, first they were plasmatized. Now they will be plasmolized. Plasmolized. Moptized, swelling up. Okay. Plasmolized. They'll shrink.
When you add salt, when you become isotonic now?
Too much. Too much, yeah. Yeah. Too much, yeah. Shrink, shrivel their cell wall. Cell membrane will shrivel. Yeah. I think I asked this question to the class at 11 o'clock. Not everybody's in 11 o'clock class. So if you were in that class, don't ask this question. Don't answer this, please. Okay, I asked this question. Name a bacterium that will crenate in hypotonic solution. Okay? You already answered that question. Okay. Name a question. Name a bacterium that will crenate in a hypotonic solution. Yeah. Name a bacterium that will crenate in hypotonic solution. Okay. Yeah. Please do.
It would be mycoplasma.
All right. Mycoplasma. Okay, let me write it down. Okay. Mycoplasma. Mycoplasma. Right? Yeah. Right? You already know that, Chris. He was there. Mycoplasma. Right? Okay. Okay.
Oh yeah, I forgot what you said.
Crenation,
Because you said
hypotonic.
it's only hypotonic.
Cells do not crenate in hypotonic solution. They swell.
Professor, could you explain why
Okay.
when you put too much salt in your or anything that you swell your body, even when you're like trying to get in shape, they say cut it up with the salt because it swells your body. It makes you retain water. The
Yes.
salt.
Yeah. That's why.
That's the case of hypertonic solution.
Yes.
Because the cells are crenating and they're giving away their water.
Water. People who are hypertensive should cut down salt because salt gains water. Salt retains water.
Salt, no, and then what is happening is that the cells are giving away their water inside,
Mm-hmm.
and that's why there is too much water, not outside.
Mm-hmm.
How do you do that? So I just.
No. Go to Dr. Siddiq. He will explain it very good to you. No, please. Yeah, he's a doctor. I'm not a doctor. He will explain it very good, really. Yeah, please. I'm not a doctor. He is. Yeah. All right. Seriously.
So salt makes you gain water.
Gain
Gain water. That's what you said.
Salt makes you gain water. Yes. Yes. That much I know. Why? He will explain it to you much better. Yeah. Oh, sure. Yeah. Okay. Sorry.
It's just like what if you did AP2, the aldosterone. If you have a low blood pressure and you want to increase your blood pressure, you're going to release the aldosterone, which is going to pull back water. So urine is going to hold back water.
Microbiology, please. Thank you. Okay. All right. All right. All right. What else? So let's come back here. Do you understand this now? She does not understand, because my question was, name a bacterium that crenate. Crenate means shrinking, shrink in hypotonic solution. And cells do not shrink in hypotonic solution, they swell. So the question was contradictory. The answer is none. None. Okay. The answer is none.
I said, is it possible?
No, none. Now you understand it? Okay, very good. Now the same question, if I asked you, name a bacterium that swells or gain water in hypertonic solution. The answer is none. Lose water, don't gain water. So please, as I requested before, read the questions very carefully on my test. Okay? Because I do. Yes, I do. So I'm just telling you. So please read the questions very carefully. Okay? More questions, please. Flagellum? Sure. Sure. Okay. Flagellum. Who has it?
No.
Gram positive, gram negative, flagellum. Okay. Both. Okay. Both gram positive and gram negative have it. Okay. Made up of what? Flagellin. Okay. Both gram positive and negative bacteria, they have it. It's made up of flagellin. Okay. Arrangements. Okay. It could be polar. Polar. 2, 3. Thank you. Polar. Monotrichous. Mono. Single flagellum at one end. Lophotrichous, more than one at one end. Lopho. Or amphi. Both end single or more than one. Okay. Or peri. Peri. Okay.
To the last one, the example of a peri.
E. coli looks like that. Salmonella looks like that. Yeah.
The two last one are the peritrichus?
Yeah.
Okay.
Yeah. And axial filament. Spirochetes have them. Okay. Spirochetes, yeah.
Axial or endo?
Axial, yeah. Endoflagellum, yeah. Borealla brugdiferi and Treponema pallidum. Yeah. Axial filament. Okay. Or endoflagellum. These are all exo. This is endoflagellum. Okay? Yeah. Yeah. Spirochetes, only spirochetes have these. Spirochetes. Okay. There's one more thing that came to my mind about flagella. Pardon me? Not the pilots, no. Yeah,
The
no.
mono, lopho, and amphi are exofilament.
These are all exo. All are exo right here. These are all exoflagella. Membrane. Yeah, they are embedded. They have parts, remember? These have 3 parts. Number 1, filament. filament hook. Yep, hook. And number 3, basal body. Basal body. Okay. What else about flagella? Rotation, full rotation, not wiggle, full rotation. Yeah. Not mortality. I'm sorry. Motility. Motility. Yeah, movement.
I got a question right here. Endospores are resistant to structure due to eukaryotic acid and mycolic acid in their spore coat. I thought they're made of only eukaryotic and calcium.
That's why that choice is wrong.
So that is the only one wrong.
No, there's another one that's wrong.
A and B
Are wrong.
bacteria, okay, good.
You need to take some salt.
Oh, I put it down.
Yeah. Okay. What next? I did not hear you. Prokaryotic and eukaryotic differences? And yeah, let's list them. Sure. Prokaryotic and eukaryotic. Differences and similarity. Let's list them. Prokaryotes and eukaryotes. Before I do that, let me ask you one question. So you just came back from a vacation somewhere in Brazil, and you collected some water from some reservoir, okay, during your vacation. And here are microbial analysis of bacteria from that river. These bacteria, they have protein, lipopolysaccharide, no sterol, chromatophores, and pylene. Okay. Is this bacterium gram-positive or negative? Gram-negative. What tells you that? Two things. Gram-negative. Gram-negative. Is it autotrophic or heterotrophic? It is autotrophic because it has chromatophores in it. Autotrophic. Prokaryote or eukaryotes?
I would say pro because we know sterols. I would say pro.
Prokaryote. Okay. Possible pathogenic or non-pathogenic?
Can you tell from that?
I wouldn't say it's pathogenic.
What has protein? Protein.
Pathogenic, I would say
I'm gonna
no,
say-
because it has the lipopolysaccharide, which is the antigen presenting that
Pathogen to toxin.
is pathogenic toxin.
Remember, lipopolysaccharide has two functions, antigen and toxin of bacteria. So potential pathogen here, and potential pathogen here, capsule. Yeah, because this is a little doubtful, but this confirms it right here. Because capsules are made up of protein. But other structures are made up of protein too, like fimbriae and other. But this should tell you right there.
That
Yeah. Lipopolysaccharide.
Makes it
Pathogenic, virulent, yeah, pathogenic, yeah.
Can you do some more like that? 'Cause that's unintensive.
Yes. Yeah. That's why before I did prokaryotes and eukaryotes, I wanted to give you this. Yeah.
Something like that, we're gonna have to label it.
Not label it, just pull your hair out. Yeah, yeah. Now, just about 4 questions are like this. 4, 3 or 4. Yeah. So now let's do prokaryotes and eukaryotes, then I'll come back to that. Okay? Yeah. All right. Prokaryotes. Eukaryotes. Okay. Prokaryotes are ribosomes. Oh, first of all, let's start with the major one. Nuclear membrane. Nuclear membrane. Present, absent, right? 1. Number 2, ribosomes. ADS, 70S, right? Three, histone proteins. Histone proteins. No. Sterols. Sterol lipids. Okay, present, absent. Okay, what else was there? Oh.
Reproduction.
Oh, reproduction, yeah. Reproduction. Reproduction. Both asexual and sexual. Asexual. binary fission, binary fission, binary fission.
You could put in organelles.
Yes, organelles. 6. Organelles. Yes. No. Okay. What else? That's about it. Do we have class here or no? No, I don't think so. Maybe 4 o'clock. Yeah. Yeah. Let's practice one more question like that. Yeah. All right. Okay. Okay. Again, you'll have a story. And these are the characteristics of the bacteria. Glycoprotein, no pylon, no chromatophores, no flagellin, peptidoglycan present, and teichoic acid present. Is this bacterium gram-negative or gram-positive? Positive, G positive, only in gram-positive. Very good. Okay. Is this bacterium motile or non-motile? Non-motile, no flagellin. Yeah. Autotrophic, heterotrophic. Very good. Heterotrophic. Pathogenic, non-pathogenic. Pathogenic. Capsule. Capsule. Capsule, glycoprotein. Capsule. Not any type of protein. Fimbri also makes it pathogenic, allows the to stick, like gonorrhea. So fimbri protein, yeah. Yeah, fimbri. So fimbri will make it pathogenic too, fimbri, sure.
I thought
Glycogen?
No. Forget it. Peptidoglycan.
Peptidoglycan?
The peptidoglycan.
Peptidoglycan I just threw in, put in there to throw you off. Because, no, seriously, I did. Because peptidoglycan cannot let you differentiate between gram-positive and negative because both of them, they have it. So, yeah, exactly. I didn't put less or more, yeah, exactly, yeah. Exactly, right. Yeah, exactly, right. No pylon, yeah. So these two tells you it's gram-positive, this and that. Positive, no pylon, positive, yes. Good day, guys. See you Saturday.